So hello everyone. We're going to start our presentation today. So thank you for registering with Fujifilm presentations and webinar. Um, Today we're going to talk about the vitrification, and if you have anything to share with us or question, please use the hand, simple to speak. If you prefer to type your question or your comment, that's fine. So we'll start talking about the vitrification, but we specifically going to talk about one thing or one factor that is affecting the vitrification process. So we know vitrification has become like a routine practice in every lab. This is worldwide now, and we all know that we know that the technique, it depends on many factors. But to achieve the successful vitrification, we need to appreciate the cell stage that we handling, the skills and the competency for the practitioner, device, and lastly, the media. And this is what we're going to talk about today, media only. So we're not going to discuss the process or the device or anything about the factors that might affect the vitrification process. So we're only going to talk in this presentation today about the vitrification media only. But in general, we know that the vitrification, it's all related about the dehydration of the cell, and we need to reduce the intracellular water, and then we need also the rapid cooling rate to achieve the glass transition. And when we do the warming, we have to do the reverse steps and to bring the cells back to active physiology status. So for the vitrification media, we know that the vitrification media was not commercially available before. And the vitrification started with animal models. So vitrification media was before made in-house. And it's made based on the understanding the theory behind the vitrification. So in many laboratories, they were using the in-house made media before, but then the commercial media started to be available. And from the studies, we know that the vitrification needs high concentration of the cryoprotectant. So it's based on that the permeable cryoprotectant needs to replace the intracellular water to achieve a good glass transition, and the non-permeable cryoprotectant to reduce the intracellular water, and also the macromolecules, which is kind of non-permeable cryoprotectant, is playing a role in the same thing to prevent the osmotic shock and to stabilize the membrane. So for the proper vitrification media, we need also to use buffer, and the buffer might be HIPs or MOPs or both of them together. So when you use both of them, you definitely reduce the concentration of one of them. So both together, it might be less in concentration than both, and that is reducing the effect of one high concentration buffer. And to add all these components or the ingredients, we need the solvent that we call the base media. So any vitrification media needs the base media, and then you start to add your all ingredient. So the media needs to be high viscosity and to provide all what we mentioned about the permeable and non-permeable cryoprotectants, and using the macromolecules to actually help the theoretical principles of the vitrification to be applied with any cells. So We talk about the base media, and the base media usually is something that we need to use for support, is expected to support the physiology of the oocytes in the embryos, and also providing the necessary nutrients. And from the history, there were many base media that have been used with vitrification, in between like simple media and more complex media. And the three is like M199 CSC and the minimal essential media. It's just an example. There is more different kind of media that is being used as a base for making vitrification media. So what we expecting from the media again is just to support and to be suitable to be used for embryo and oocytes. The composition of the basal media, it might be, as I said, it's simple or might be complex. But what we can see is always that we always see in the composition salts and energy substrates, amino acids, and a buffer, and that might be the bicarbonate buffer, like in cultural media. And whether the phenol red is present with the media or not, we know that the phenol red does not have a necessary or certain function in the vitrification itself. So absence of the phenol red or the presence, it's not going to affect the process itself. So this is what the basal media, and it's important to choose the basal media that is actually supporting the kind of cells that you are using. And now we move to talk about the cryoprotectant. So the cryoprotectant is the main active component of the vitrification media. And the concentration of the cryoprotectant is exactly what is helping to transfer to solid status and to transfer the fluid to a glass format. So we know that the cryoprotectant is, there is two kinds, is permeating cryoprotectant and non-permeating cryoprotectant. So the cryoprotectant, it's the permeating one is known as to be low molecular weight, and the other ones other kind of cryoprotectant that is high molecular weight. So the cryoprotectant is used to protect the cells against the cryo injury, and that is by replacing the intracellular water. And the non-permeating is protecting against the osmotic shock. So both of them, they play a role to protect the cells from the cryo injury and reducing this effect. But we know also that the cryoprotectant has cytotoxicity and has this effect. And this effect might be caused by osmotic shock or biochemical modification of the cells. And both effects, that is actually what is the main reason for cryo injury and that cell damage. And we know also that the toxicity is related to the concentration. So we need to find the balance between the concentration and the cytotoxicity. And this is what the studies long time ago were studying, how to achieve the aim and to do a successful vitrification with minimizing all the toxic effect. So the balance is to reach your glass transition with the minimal effect of toxicity. So the permeating cryoprotectants, this is just an example. There is more actually can be used for the permeating cryoprotectant, but the most common that being used is the DMSO and ethylene glycol, and the propylene glycol, and the propanediol, and the glycerol is the least that it was being tested before, but because of its glass transition and related to the high temperature and the rate of the cooling, is less commonly used in vitrification of oocytes and embryos. So the permeating cryoprotectant, as I said, it's the low molecular weight, so they can permeate the cells and they can replace the intracellular water, and also it's non-ionic with high solubility in the water, so that reduces the freezing point of the water. So the permeating cryoprotectant, these are the main cytotoxic in the vitrification solution, because they are able to enter the cells, and with the interaction with the intracellularly, that is what sometimes cause also again the cryo injury and cytotoxicity. And when the permeating cryoprotectant enter and replace the water intracellularly, that also reduce the ability of any ice formation later on during the vitrification process. But with the studies and with the history of studying all the vitrification process with different kind of cells, and also from the animal studies, we get to learn a lot about the cryoprotectant and the relation how to reduce the cytotoxicity. So we know that the cytotoxicity is mainly related to the concentration of the cryoprotectant, and every cryoprotectant has a certain need a certain rate for cooling to be transformed to glass status. And so we found that the combination between two or more than one cryoprotectant is helping to reduce the concentration. And when we reduce the concentration, we reduce the molarity. And that is all will help to reduce the cytotoxicity. So lowering the molarity, it means that we're lowering the cytotoxicity. And we also know that the toxicity is time and temperature dependent. So we get to learn that the cells are more permeable to cryoprotectant at a higher temperature, but it gets more resistant to the toxic effect at a lower temperature. So this is all related to what we use to balance adding the cryoprotectant with avoiding the cytotoxicity effect. So the cytotoxicity effect also is related to time. So when we try to vitrify in a high concentration of cryoprotectant, we need to be really careful about the time. So from the studies, we reached the point that we can dehydrate the cells in 2 steps rather than 1 step, and to what we call it now, the equilibration period, and then the vitrification period. So you can spend more time with lower concentration. So that is allow the cells to be dehydrated properly before the rapid dehydration and before to be exposed to high concentration of cryoprotectant. So that is all revealed from the studies that is helping to improve the results and improve the cell survival. We also get to know from the studies that every cell stage is different. So we know that the movement of the water and the cryoprotectant, the permeating cryoprotectant through the cell membrane is different, although it's very important to understand, and is one of the crucial things that is affecting the survival during the vitrification process and during the cryopreservation. So the degree of the permeability of the permeating cryoprotectant is important to understand. And as I mentioned before, it's also related to the temperature. So the oocyte is different than the embryos because every cell stage has a relative surface to the intracellular water volume, and that is one of the things affecting the permeability. And the method of permeating is also different. So every cryoprotectant has a different mechanism to enter the cell. So some of them, they enter faster, some of them by simple diffusion, and some other it has to be through channels. So all this needs to be understood well, and to understand how to expose the cells to the cryoprotectant and appreciate time and temperature and concentration in the same time. So again, the aim is to have the permeating cryoprotectant to replace the intracellular water with the minimal effect of the toxicity of this cryoprotectant. So that is the improvement that has happened through the years. And we now talk about the second part of the cryoprotectant, which is the non-permeating cryoprotectant. So we can't use only the permeating cryoprotectant only. It's the presence of the non-permeating cryoprotectant is also supporting the process and the vitrification itself, although they can't enter because of the high molecular weight, but they play in a different way to protect the cells. So with the non-permeable cryoprotectant, like we have sugar and we have macromolecules. So the most common sugar used in the vitrification, the sucrose being used for a long time, and triulose, raffinose, and mannitol. And this is just an example. There is more and more sugar that is actually being tested and being used to make the media when it was in-house made. So this disaccharide is actually playing important role in stabilizing the membrane, and it interacts with what we call the polar head groups of the membrane phospholipids. So it has a kind of chemical interaction that is protecting the cell membrane, and it helps to increase the tolerance to the freezing. So the macromolecules is non-sugar, but it's different kinds like dextran and serum albumin, fecal, and there is other synthetic macromolecules can be added. Again, the macromolecules is playing an important role like sugar, and the concentration of altogether the permeable and non-permeable cryoprotectant. This is what creates the viscosity and the concentration of the fluid that it needed. It's important to have this viscosity and the concentration and high concentration fluid to be easy to turn to a glass transition and to turn to solidified status when it's plunged to the liquid nitrogen. So adding the non-permeable cryoprotectant, it's also reducing the molarity of the permeating cryoprotectant, So it decreases and reduces the effect of the cytotoxicity of the permeating, and also it increased the osmolarity in the extracellular space. So that also enhancing the dehydration and reduce the intracellular water. So when we reduce the intracellular water, it should be less risk of ice formation. I just have to say that the complete 100% dehydration is something difficult to achieve in cells. So, but we try to minimize as much as we can the water content in the cells, so we reduce the ice formation. So also addition of the non-permeable cryoprotectant to the warming solutions during the warming process is helping to decrease the traumatic cell expansion during the rehydration, and it's also to help to stabilize the membrane. So that's exactly the function of permeable and non-permeable cryoprotectant. But we will talk a little bit more about the sugar that we add, and sucrose was the most common used one, and it's very efficient to be used. But recently, the triulose use is become more and more. And it's because of the triulose has a higher glass transition, and adding the disaccharides in general, it's decrease, as I mentioned, the molarity of the permeating cryoprotectant and decrease the effect of the cytotoxicity. And another advantage of triallose that it has a stable glycosidic bond, and that make it different than the other sugar. So we know that the triallose is one of the naturally occurring sugar, and it's found to be the blood sugar in insects, and we found also that it's being present in the natural frozen creature, that some insects that they have to go through freezing during winter and tough winter time, and then it happens that they thaw, and they use in nature more glycerol and triallose, because they have the triallose in their blood that is helping also to go through the whole winter and survive it at the end. So the triallose, again, it stabilizes the membrane, and it also has poor solubility at low temperature. So it's stabilizes the protein in the solution of the vitrification media. And it helps to reduce the crystal, because it's interacting with water more than other sugar. So this is, again, all the components that we need to vitrify with any media, and the same what all laboratories when they use the try to make the vitrification media in-house, that's the same principles that they use. But we have more commercial media now, and the vitrification theoretically all based on the same principles. So most of the media, they use the same principles and the same ideas to make the media. But not every lab not all the laboratories, they use the same media. So every lab, they have different media, and all over the world, sometimes we don't have certain media available. So sometimes in some certain regions you find media available, but it's not available to other regions. And it happened also that embryos and oocyte transportation is more available now. So we can ship the gametes and embryos to anywhere now. So it comes to the point that the embryologist might think that, oh, what I need to do if I receive embryos being frozen in a certain media, that I don't have this media available. And so hence comes the idea of trying to thaw embryos and oocytes in a different media than the original one that is being used in freezing this media. So it's interesting studies that try to see how is the cross thawing affecting the survival of the cells. So one of the studies that is for Ludovico Parmigiani, that this study is actually took a long time because it has a high number of cases, and it uses two different media, and it shows how is the thawing process as a theoretical principles to apply it, and using the non-permeable cryoprotectant can be actually applied with different media, and it can be applied safely, and you still can get the whole results and the same or maybe better results. It did not even affect the pregnancy and the development of embryos. And another study that is also recently studied, that even if you use media with different base media, so even your base media is a little bit different, that did not affect, again, your results. So the idea of using different media than the originally being used for freezing is not that risky, but I think we need more and more studies to see how is the cross thawing between all available media in the market is possible or not. So that was the whole talk today. So thank you very much for listening. If you have any question or Any thoughts to share that you are welcome to share it with us? So we have a question actually said, if we have frozen in different media, can we use your media? Right. If we were to generally, it happens that these studies, it showed that you can use Irvine scientific media for thawing in a different, and even I know that also there was one paper that, or maybe more than one, tried to use Irvine scientific media to thaw also the slow frozen embryos. So the whole idea of the theory, it can be applied, but it needs a little bit of courage to apply it in your lab. So if you have studies between two specific media, then you feel like, okay, I'm really willing to try it in my lab. But if there's no studies, then I think anyone would like to apply the idea in the lab. It has to be under your responsibility, right? It's not something that we can do for a clinical application. I think it needs to be for an experimental trials first, and to see whether this media going well with this media or not. But again, in the non-permeable cryoprotectant, I don't see the conflict for thawing any other media. The point is, again, it's not only about the media. So when we started the presentation, we started to talk about the vitrification is actually related to many factors. It's not only one factor. So you get to know that if you would like to apply and try the idea of using one media and throwing in another media, you just have to go through all the factors. Check your device, check your vitrification rate and warming rate, and all this is affecting. So if it did not work, it does not mean that they can't work together or they cannot have a cross matching or cross thawing. It's all, how did you see, and your prospect, how did you see the whole idea? You need to go deeper and deeper in the whole factors that is affecting the process itself. So there's no more actually any more questions. And thank you very much for listening. And again, if you have anything to share with us, or your thoughts, your ideas, if you would like to try in your lab in two different vitrification media, I would just, the last thing that I would like to say, that you need really to look properly what you use in your lab, because your media is telling you what to do for the concentration, for the temperature, for everything. So it's closely related. So the composition of the media is actually deciding how you should use this media in your lab. So that is something important. Well, I think we came to the end of the our session. And thank you very much again for listening. And we may see you in the coming webinar. Thank you very much.